Alright, so now getting into the technique portion for today's lab. Today we will be looking at some of the plants here that we brought in that have symptoms and signs of infection. We're also going to be working with last week's plates that you guys inoculated with your bacteria, or what we hope, bacteria if you didn't contaminate your plates. And we're going to be doing step 2 of Koch's postulates. So can you guys remind me, what is step 1 of Koch's postulates? Observe, right. You got to observe the pathogen, the suspected causal agent in every single one of the infected, in this case, potato slices. So then the second step is to isolate it on a pure culture. That's what you guys are going to do today. If everything went well in your boxes last week, you should see a bunch of different types of bacteria and fungi on your plate that grew over the week. And the reason we did the whole quadrant streaking was so that we would be able to isolate those, because if you guys just took your inoculating loop and rubbed the entire plate, all the microorganisms would be growing on the plate. You wouldn't be able to distinguish between one and the other. You wouldn't be able to have a pure culture, and therefore you would not be able to fulfill Koch's postulates. So that's the reason we did the quadrant streaking. So now that we have, now that your plates have grown, we're going to isolate those bacteria or fungi by taking your inoculating loop and just touching. You don't have to rub the colonies, just simply touching the loop to the colony and then streaking another plate is enough to transfer it. And so we're going to try to be growing the microorganisms onto a new plate. Alright, so that's what I just said. Some general characteristics of bacteria is that they're unicellular, unlike animals who have cells working together. You do have some bacterial species that do work together, similar to, if you guys have taken your microbiology classes called quorum sensing, in which the bacteria are able to interact together. But for the most part, each cell of a bacteria is able to grow on its own, whereas if I were to take one of my skin cells and start growing out of the plate, or separate it, would not grow by itself into another one of me. So that's what is meant by unicellularity. They're also prokaryotic, so they don't have any. They don't have a nucleus. They have a rigid cell wall, and we have different types of cell walls between the different types of bacteria, and we'll showcase that next week. No membrane-bound organelles, so everything's, for the most part, floating around. Also, they have circular DNA, meaning it's just one big plasmid for their entire genome, whereas animals, they have telomeres at the end because their DNA is linear. They also reproduce through binary fission, so it's a little bit different than the type of replication or reproduction that our cells do. But I won't get too much into that since we won't be asking you guys about that. They also reproduce a lot faster than animal cells. So within 20 to 30 minutes for E. coli, you can already have a new generation. Whereas with humans, it can take up to several days. There's also different types of shapes of bacteria. And again, we'll be going over this next week, and when we actually look at the bacteria under the microscope, when we look at the individual cells. There's flagella, which are little tail-like organelle or structures on the outside of the cell that allow the bacteria to move. And for signs, when doing the diagnostics portion of the plant pathology portion, we look at the exudates or the ooze, which is what we saw in one of those images, streaming. And then bacterial ooze is when you see, if you've ever seen one of these trees on campus start to kind of like, it look kind of like sap, but it's very dark, just start to spill out of it. That's actually bacterial ooze. See, they multiply rapidly. Some bacteria have enzymes or toxins that destroy parts of the cell wall of plants. Recall that plants have a very tough cell wall, and it's difficult for the bacteria to get inside of the cells where all the nutrients are. So what they do is they release these enzymes that start to chew away at the cell wall, make it soft and weak, until eventually it bursts open. That's actually what a lot of bacteria do to cause it to rot. When cells start to open up of a plant, you start getting that browning. If any of you have ever eaten an apple, probably most of you have, if you take a bite of it and then you just let it sit there for a while, the apple starts to get brown. The reason it gets brown is because you broke all those cells by chomping into it. And now all the enzymes and stuff are starting to leak out and starting to rot. So what the bacteria are able to do, they're able to break down the cell wall, not by chomping into an apple, but by chemically degrading the cell wall. So that's pretty cool. And then they can also, some bacteria can also cause gall formation in some plants. And then for next week, we're going to be doing the step 3 of Koch's postulates. What is step 3 of Koch's postulates? Right, re-inoculating. So today we're trying to get pure cultures so that next week we can start inoculating those pure cultures onto bacteria so that we can look at step 4, which is to see if we can get the same symptoms or the same symptoms as what we saw in week 1. So what we're going to be doing today then is you will take, you guys should have 2 plates in each of your boxes. You're going to take your inoculating loop. Just hang on a second, listen to what I have to say. You're going to take your inoculating loop, just touch one colony of whatever you have growing on your plate that you streaked on last week. And just do that quadrant streaking technique again. Make sure everything's sterile, make sure you don't contaminate anything. If you contaminate anything, I've got extra plates here, so just let me know. And if you do everything correctly, then you should have pure cultures for next week. On top of that, you will also be recording the symptoms and some of the signs of these infected plants that we have in here. That'll be for your assignment number 3, I believe. Yeah, assignment 3. So for you guys that are online, your assignment 3, you will still have to do assignment 3, but we'll upload pictures of the things that we have here in the lab today. And I believe that's it. Yeah, so your plates should look something like this in which you have individual colonies. Just touch whichever one you want. Make sure you label it. That's something that I forgot to mention last week. When you have the bottom plate, which is the plate that has the actual media on it, when you write your name on it, make sure you write along the outside, because if you start writing on the center, you'll be blocking your view of some of these colonies. So just write your name on it. Again, when you finish it up, put the plate upside down inside of the bag that's inside your box, and just put that in the box and you're free to go. Any questions before we start? All right, get to it.